So, listen and take notes from your online lectures, attend the fortnightly lectures, revise, ask questions. Anything that's not clear, please contact me. Make an appointment with me. So I have a lecture now at 12 o'clock, but that means most Wednesdays I get here early and I work on campus. So I'm available Wednesday mornings. So make use of it. Don't fall behind with your weekly online lectures, and don't leave your study until StuVac. Please don't. And you really do need to keep up with this content. Okay, so that's really an introduction to how the unit runs and what's it about. Any questions from what I've said? No? Okay. So let's get started on genetics, genes, and human variation. All right. How many base pairs of DNA are there in the human genome? Yes. I got the same answer yesterday, so I've got to improve the way I asked that question. You're absolutely right. There are 4 bases. What I really wanted to ask with that question, though, was how many bases are there in the human genome? So how many base pairs in total are there? Not different ones, but in total. That's the answer to another question on this slide. Yep. It sure is. 3 billion in a haploid genome. So 3 billion base pairs made up of 4 different base pairs, A, G, C, and T. You're right. Okay, so we've got 3 billion base pairs. Now I'll ask you your question. How many genes are there in the human genome? About 25,000, that's right. That's our current estimate of the number of genes. It keeps changing because we're still discovering lots and lots and lots about our genome. And the reason we are is because we are now able to analyze the genome in its entirety. The actual number of genes is likely to be somewhere closer to about 30-odd thousand. That 25,000 number really represents the genes we know about in terms of protein coding genes and genes that only make RNA that don't make protein. Can someone give me an example of a gene that only makes RNA? You would have studied it in functional proteins and genes, molecular biology. It combines with a protein and it's important for translation. What sort of RNA am I talking about? Ribosomal RNA is one, and another one that sort of connect the ribosome, but you need something else to sit on the RNA and read. What's that called? Transfer RNA. Good, thank you. They are structural RNAs. They are made by genes that make RNA but do not make protein. There are lots more genes that make only RNA, or what we call regulatory RNA. Regulatory RNA changes gene expression. It can regulate the stability of RNA. We've got microRNAs, all sorts of things. And we keep discovering these things. So that's why I say we've got about 25,000 genes that we are really certain about. But with all these new RNA coding genes, it's likely to be somewhere much higher. How many chromosomes make up the human genome? Yep. 46 in a diploid genome. Yep. And they are made up of autosomal chromosomes and the XY or XX. Is my DNA identical to yours? In what way is it different? Saw the shake of the head. You're absolutely right. It's not the same. How is it different? Let's leave XY aside for a sec. Yeah. Mutations, yes. How many genetic variants do you think we have in our genome? At least 4 million. And some populations 5 million. And that's not counting insertions, deletions, and all sorts of things. It's full of variation. But within a 3 billion base pair genome, 4 million variations is not really that much. But they're the variations that we're going to be looking at in this unit, because they're the ones that influence human health. So no, my DNA is not identical to yours, but I share a lot of the features of my DNA with the population that I belong to, which is Caucasian, Northern European. Of course, I'm most related to my family, but then I'm most related to my origin, my population that I come from. A lot of what we look at in this unit is all about populations. Where did we come from? Where are those populations now? How do populations differ in their genetics? And what implications does that have for the health and susceptibility of those populations to certain diseases? So this is really where genetics started, way back in Mendel. And you've all heard about pea plants, probably started in high school, you've probably heard it in first year university. And what Mendel did was really show in his pea plant experiments the first laws of inheritance, which is, if we look here, that he had his pea plants. Of course, he didn't know about this, but he did see this, that purple, white, F1 is purple, F2, they segregate in predictable ratios. What he was looking at was genetic variation. And he was looking at features such as dominance, recessive alleles, codominance, all of these things that you've probably heard about in various units that you've taken. So that's the basis of genetics. It really says, put it in really simple terms, although nothing in genetics is really simple, but if I can be simple, what I will say is that Mendel, in its simplest, says you've got a gene, it has a variation, and that variation has a measurable effect on a phenotype. We see that in the classic Mendelian diseases. Does anyone know a Mendelian disease in the human population? What is an example of a Mendelian disease? What it means is it segregates as Mendel would have seen. It's predictable segregation of phenotypes. Yes. Yes, it would. Yes, absolutely. Very rare mutation, but yes. Cystic fibrosis is one. Huntington's disease is one. There's actually heaps and heaps of them. We'll have a look at a couple. So what we need to understand is homozygotes, heterozygotes. This is the two chromosomes. We each have 2 copies of a chromosome. Why do we have 2 copies of each chromosome? Why? Why do we have a diploid genome? Where do those 2 chromosomes come from? Yes, mum and dad give us 1 chromosome each. In other words, we get genetic material from 2 different sources and we bring that together. That's part of genetic variation. That's actually independent assortment way back in the gametes of mum and dad. So you can see here that I've got 2 chromosomes, 1 or 3 sets of 2 chromosomes. This one has an AT base pair. This one has an AT-GC. This GC-GC. Homozygote, heterozygote, homozygote. The way we would write that is that this person here would be an AA homozygote, an AG heterozygote, and a GG homozygote. In genetics, what you will see is we don't write the base pairs, we just write one of the bases. And it's usually from the upper strand or the coding strand of the DNA. Because if I write you're an AA, it's assumed. You know that we're AT-AT because of complementary base pairing. So that's the first thing, and you'll see that in the computer classes. When we write DNA, most of the time we just write a single strand of DNA. You don't need to write both strands. This is what we call a karyotype. This is the human genome, the 22 autosomal chromosomes plus XX, XY. Each chromosome has its homologue, you can see here, and you can see that not all chromosomes are equal. Chromosome 1 is the largest, most gene-rich chromosome that we have. We move down to chromosomes 21 and 22. They are the smallest, least gene-rich chromosomes. They actually don't even have a functional what we call P-arm, this arm here. It's all what we call heterochromatin. Does anyone know what heterochromatin is? Have you heard that term before? It's DNA that is tightly packed. It is scrunched up. We will be covering a lot about heterochromatin. The opposite to it is something called euchromatin, which is more open. Structure of DNA is really important in this unit. So chromosomes are different, and of course there are chromosomal abnormalities that are not common but not infrequent. Down syndrome you probably all know about. This is trisomy, where someone has 3 copies of chromosome 21. That is one of the few, with the exception of Turner syndrome, one of the few human genetic abnormalities that tolerates more than the normal number of chromosomes. If you had a fetus with, for example, 3 copies of chromosome 1, that would not be compatible with life. The reason this is because it's such a small chromosome and it's relatively gene poor. Of course, there are developmental and intellectual disabilities that go with that, but they can live a life lasting many decades. There's translocations. This is a deletion, Turner syndrome. With females lose an X, you can also gain XXY. So the real question in genetics is, what does it mean for phenotype? We've talked a little bit that our DNA is not the same, that we carry genetic variants. When we look around this classroom, we see genetics in action. By our skin pigmentation, hair pigmentation, height, probably if we look deeper into blood pressure, other measures of metabolism, you would see differences that reflect our different genetic makeups. Of course, it also reflects diet, and it can also be cultural as well. But what we see around us, the variation, is first and foremost genetics. And genetics is all about understanding how does that variation translate to phenotype. With Mendelian genetics, it's relatively easy. You can see a mutation, and that mutation is correlated with a phenotype, such as achondroplasia and the short stature. We are not going to be looking a lot at Mendelian genetics in this unit. Matter of fact, very little. Because what genomics is doing, it's moving away from Mendelian and starting to ask different questions. It's not to say that Mendelian diseases are not important. They are incredibly important. But the genome and the understanding of the genome offers us the opportunity to look at common, complex diseases. These are the big diseases that are starting to affect our society today. Can someone tell me what I might be talking about? What sort of diseases do you think I'm thinking about right now? If I was to ask you, read the newspapers, tell me some of the big health issues that are facing Western Sydney. It's been in the newspaper. Can you think of a disease the last 3 or 4 months? They're talking about it almost in terms of an epidemic. What am I talking about? This is your own backyard. There's a big, big health thing going on. What is it? Exactly. You've got it. Diabetes. Spot on. It's a special, but it's not all diabetes. It's called type 2 diabetes or adult onset diabetes and some other things. And what's type 2 diabetes associated with? Obesity. Yes. Lifestyle. Okay? It is being called a health epidemic. Not just in Western Sydney, although there has been a lot of coverage of that. Our school actually does a lot of research into the socio-economic factors of that, and there's lots of people trying to understand the genetics of things like that. Obesity is a metabolic disorder. It's also a lifestyle, but it is. You'll get some people who'll be able to eat this food. Another person can eat the same food, obese, non-obese. So there is a genetic component, okay? It is a complex disease. There are many genes involved. Those genes carry genetic variants, each of which contribute a very small amount to the phenotype. And there's a big environmental input. Understanding these complex diseases, which have a genetic basis and which are of high importance to us as a society, is really what genomics the promise of genomics, if I can put it that way. And it's moving beyond the idea of Mendelian genetics. So this is a classical look at a Mendelian locus. A locus is one of those jargon words. A locus can mean a chunk of DNA. It can contain one gene, it can contain 3 genes, it might contain a regulatory bit of DNA. It's just a bit of DNA that we've found that does something. So here is a locus, and it carries a gene, for example, with a mutation, and it has your 2 homozygotes that we saw earlier, and a heterozygote. Now, depending on whether it's dominant or recessive, they will segregate in certain ways. Perhaps this is the phenotype, no phenotype, and this one here might be an intermediate phenotype, or may not show a phenotype at all. It really depends on the nature of the allele, which we'll discuss in a minute. So that's really the classic Mendelian picture. An example of that is Huntington's disease. Huntington's disease is a devastating neurodegenerative disorder. It usually starts around midlife, and then it's a decline thereafter. This is a pedigree. Huntington's disease is what we call a genetically dominant disorder. In other words, the phenotype appears in every generation. It affects males and females equally. It's an autosomal disease. In other words, it's not located on an X or Y chromosome. And approximately half of all offspring are affected. That's Mendel. That's Mendelian genetics. Predictable transmission of a phenotype. Of course, it's also statistics. So if you look at that pedigree, you might say, hang on, there's more than half of the children affected. That's because each birth is an independent event. But if you looked at 100 families, the statistics would come out as I've written them here, or very close to it. So, genetics is all about probability. It's all about statistics, particularly when we start looking at something as complex as obesity, or asthma, or multiple sclerosis, or arthritis. We start talking in terms of odds ratios and probabilities and things like that, which we'll come to as we move through the unit. So let's look at Huntington's disease. This is the gene. I think it's chromosome 1 from memory. This is where, and it has this thing called a CAG repeat. As you know, triplets encode proteins, and this particular triplet repeat encodes the amino acid glutamine. This gene is called huntingtin, and at the front of the gene is a CAG repeat. In the normal population, there could be anywhere between about 4 or 5 and 27 of these repeats. So you may be someone who has glutamine, 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 but the person you're sitting next to has glutamine, 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 about 8 or 9 of them. Definitely not 8 or 9 of them, maybe 6, which I'll say why in a minute. In other words, it's genetic variation. The gene that carries a mutation that is responsible for Huntington's disease, that mutation is actually, in most people, a normal genetic variant. Some of us will have 3 repeats, some of us will have 5, some will 4, some will 12. That's perfectly normal. It's who we are. But in people with Huntington's disease, something goes wrong during DNA replication. This number of repeats is no longer stable, but it starts to expand. It's not a normal process, it's a pathogenic process. So it can go from 27 up to 35, and as soon as you get in this red zone of more than 35 repeats, you will develop Huntington's disease. No question. That will happen. That's what we call penetrance. Penetrance is the likelihood that a mutation will give a certain phenotype. In other words, if you have 38 repeats in the gene encoding the Huntington protein, you will have Huntington's disease. Absolute guaranteed. 100% penetrance. So here is a history of Huntington's, and it shows that Mendelian genetics is actually, it has a few complexities. So John and Amy, they've got 2 teenage children. The father, John, he becomes forgetful. He starts to get depressed. Then he develops what are called choreiform movement. These are jerky, involuntary movements, face grimacing and so on. The doctor diagnoses Huntington's disease, and he sees that he has this very large triplet repeat, lots of CAGs. Something's happened to his genome. Something's gone wrong, and it's expanded. So because we know this is a Mendelian disease, the doctor says, well, what about your parents, your grandparents, your great-grandparents? And all John could say was, well, my grandmother died at 90, and we thought she was a bit odd, but my mother died at dementia at the age of 78. So you could think, well, they're both neurodegenerative type disorders. I wonder whether that was Huntington's. But they lived to a very good age, well, more or less, whereas John is only in his early 50s. So if we look at the pedigree, there's no real evidence until, bang, along comes John, Huntington's disease in his 50s. This is showing us that as we move through generations, we're seeing increasing severity and decreasing age of onset. If we make the assumption that actually this may have been Huntington's disease, what it shows is that Huntington's disease exhibits something that we call genetic anticipation. In other words, those little repeats. In John's grandmother, something has happened to her genome where those repeats have started to expand. She's passed on that problem to her daughter, where they started expanding a bit more. By the time it gets to John, when she passes on her DNA, they've gotten even bigger. And now he has Huntington's disease. In other words, when you look at that pedigree, you'd say, that doesn't look Mendelian, but it's actually revealing the true nature of the way this genetic variant works, which is it gets worse as you go down through generations. And that's called genetic anticipation. Of course, that has big consequences for their children because he is now a carrier. That means that they have a 50% chance of being affected. The other children, of course, and their children also may be at risk, which means genetic testing is needed. Which then brings up the question, who do we test? Do we test the teenage children? Do they have a right to say no? Can we force it on them? Do they need to know? What about their children? What it's starting to show is, firstly, that Mendelian genetics is incredibly important. It's not always as simple as we think in terms of pedigrees. And secondly, by knowing something about our genetics, by knowing the link between our genes and our health, it raises all sorts of ethical questions about who should know, when should we know, and what does that mean for us? Perhaps in terms of insurance, job security, a whole bunch of things. So what we've just been talking about is a type of genetic variant called a tandem repeat, or microsatellites. There's hundreds of thousands of these. They're scattered throughout our genome. They are a relic of a human evolution. It's where we come from. Our genome is millions of years old. And through those many millions of years and divergences and everything else, our genome has undergone duplications, deletions, mutations, substitutions, all sorts of things. And so it's full of all these repeats, which can sometimes be the basis of disease. This is an example, GTAC, GTAC. It's a multiple repeat. They are what we call polymorphic. Has anyone heard that term polymorphic before? What does it mean? If I say this is a polymorphic variant, what does that mean? I saw some head nods. What does it mean? See, what you know is I like to ask questions because it keeps you awake. And by asking questions, you don't just be passive. And it makes it more interesting for me too. So, polymorphic, what does it mean? Yes, Yes. So in other words, that you see a variation in the normal healthy population. Exactly. We saw that with Huntington's, that you could have somewhere up to around about 25 repeats in the population. You may have less, you may have 25, you may have 2, 1. Yeah, that's exactly right. You see lots of different forms of that genetic variant in a population. It's perfectly normal, it's who we are, and it may or may not contribute to our phenotype. Does anyone know the earliest use that these little microsatellites was put to? You're on Hawkesbury campus. Who does forensics? No one. Yes, one. You should know. The earliest use by a guy named Alec Jeffreys. DNA fingerprinting. That's what he used. These sort of things here. We have hundreds of thousands of these things. They all have different lengths, which means they can be used as a type of barcode. And that's what the earliest DNA fingerprinting was. It's advanced a bit from then, but still, it's a way of identifying individuals. It allows you to say with a very, very high degree of certainty your relatedness to someone else. Late last year there was a brilliant television series about this, and it was really, really good. It went through the whole thing and about how it was used. The first time it was used, actually, the police came to him to solve a rape murder case in the north of England, and that's what it was all about. So it shows genetics has a very important application. Not that that's the only reason we would study genetics, but that's human variation. Okay, Huntington's disease. We've talked about the triplet repeat expansion and how it's a dominant phenotype. So what does that mean? We want to understand genes and their phenotype, but we want to understand why. How does a mutation give that phenotype? Sometimes we're lucky enough to know, other times we've really got to work at it. Huntington's disease is a neurodegenerative disorder, which means it must do something nasty to neurons. And what it does is that all of those extra glutamines, they cause the protein to misfold. It causes the protein to form aggregates. Those aggregates start killing the neurons. They start disrupting synaptic transmission. They start messing with gene transcription. And it only takes one gene to make that misfolded protein to see the effect. That's dominance. Even though these people, one of their genes is perfectly fine, that other one is making a misfolded, incorrect product, and that is leading to this toxicity and the death of the neurons. And that provides a nice link between genotype, phenotype, and why it's a dominant trait. It's not always as clear-cut as that, but that's an example. So, Huntington's disease, we talked about polymorphism. You can see that in the normal population, there is a range of repeats where you don't see a phenotype, but as soon as it crosses a threshold, you will see Huntington's disease appear. This is the gene, actually. It's called Huntington, and it's on chromosome 4, not 1. So in the normal population, the CAG repeats, they range from between 11 and 34 repeats. Perfectly normal. If we were to go into the prac next week and extract some DNA and run those repeats, that's what we'd see, okay? We're not doing that, but you could do that. So Mendelian phenotypes, they are monogenic traits. In other words, one gene. Huntington, one gene. That's to give the disease. However, the severity of the disease, the age of onset of the disease, that may be affected and modified by other factors. There may be other genetic variants which mean even though you will, without question, get Huntington's disease, perhaps you'll get it at 60 and not 50, or you'll get it at 30 and not 50. Perhaps it will be a very rapid decline, or perhaps it'll be a very gradual decline. That comes down to what is happening in the rest of our genome. Because you've got Huntington, the mutation, which is causing the phenotype, but that phenotype is modified by lots of other genetic and environmental factors. So Mendelian transmission. We can predict its transmission We use family pedigrees. We identify whether it's affecting every member of a generation, half of them, and we can start to make an assessment about what we're looking at. This is recessive, and you can see you need 2 copies of the mutation to see the disease. Even though you need 2 copies of the mutation, if you have 2 copies of the mutation, so 1 on each chromosome, you will get the disease. Therefore, it is still 100% penetrant. It just means in recessive diseases you need 2 copies, but if you get 2 copies, you'll have the disease. It doesn't mean it's 50% penetrant or something like that. It just means the rules are a little bit different. The rule for recessive is 2 mutations, disease, it's 100% penetrant. Okay? Dominant, of course, 1 copy, and you end up with the mutation. Now, what you can see here is something called a compound heterozygote, and it raises a very important point. This is cystic fibrosis. As you know, it causes a very large mucus buildup in the lungs because there's an error in I think it's a sodium transporter, chloride transporter in the lungs. The cystic fibrosis gene, when you look at a population of individuals, all of whom have cystic fibrosis, and you look at the DNA sequence of their gene, you will uncover literally thousands of different mutations in their gene called CFTR, the gene that is responsible for cystic fibrosis, or rather mutations in that gene are. So if we look here, you can see here is the defect, the type of mutation, and the mutation examples. So there's all of these different mutations. These are the locations of the mutations. These are amino acids. Phenylalanine, residue number 508, phenylalanine is deleted, and so on. We'll be looking at this kind of nomenclature in the first computer class. But what it tells us is that just because cystic fibrosis is a Mendelian disease, it doesn't mean there's only one mutation that causes it. There is one gene, but within a population, within that one gene, you will find many mutations. That is what we call genetic heterogeneity. In other words, one gene, but that one gene can carry many mutations. Those mutations may be more or less common in different populations. So, for example, if I was to go to the north of Europe, we might, for example, this mutation here might be found in 80% of all individuals with cystic fibrosis. But if I went to Japan, maybe only 3% of people have this mutation if they have cystic fibrosis. It's what we call a founder effect. In other words, if you have a population, that population will be defined by what we call a group of private mutations. They will share a lot of their genetics. And so we see in diseases like this. So Mendelian diseases, they're rare in a population. They're sufficient to cause a disease. They have high penetrance. Penetrance is the likelihood that the mutation will cause the disease. Genetic heterogeneity means that you can have a single gene, but you can have multiple mutations in that gene, any one of which will give rise to the phenotype. However, those different mutations may have different effects on the phenotype. See, it's always an if and a but with genetics. So, if we go back to those cystic fibrosis things, perhaps if you have this mutation here, you are going to have a very severe form of cystic fibrosis. Clinically, it'll be difficult to treat. Your life expectancy may be very short. But perhaps if you've got this mutation, maybe it'll be less severe. Maybe you will have a more or less normal lifespan with much less clinical intervention. That's what we call expressivity. So even though there are different mutations which cause cystic fibrosis, you will see a variation in the phenotype. An example of that is a mutation that causes, in the gene that causes familial breast cancer. Does anyone know the name of the gene that carries a mutation that causes familial breast cancer? Yes, BRCA1 and BRCA2. BRCA1 and BRCA2. It has been in the news a lot lately. Does anyone know why? There's lots of stuff about genetics. There's even something in the news this morning about microbiota and genetics and Alzheimer's. BRCA1 was in the news a lot, probably towards the end of next year, last year. It may well be next year too, but last year it's certainly in the news a lot. Does anyone know why? Something to do with patents. The company that first identified the gene BRCA1 as the gene that carries the mutations that causes familial breast cancer, the first thing they did, they patented it. They said, we own these mutations, and we own the right to make diagnostic kits to sell to women and occasionally men, because men can get breast cancer, believe it or not, and where they can be tested and make the decision on how they wish to proceed. Lots of patient advocacy groups challenged that. They said, how dare you? You can't do that. This is an essential public resource. And in the courts in Australia, there was a big to and fro about, can we still allow patents on such things? And I believe the Australian courts said yes, but then after that ruling came out, there was an equally big court case in a state court in New York that said no. So this gene and the mutation and everything was discovered way back, like about 25 years ago or something, and it's still going through the courts today. There's huge amounts of controversy around, can we patent, should we patent, who owns it? If I put $1 million into a research project and I discover these mutations, shouldn't I have the right to earn some money from it? Or is it the patient's right to have access to it? Again, this is all about genetics. But getting back to BRCA1. Mutations, again, there are lots of mutations that have been identified in the gene BRCA1. There's a particular group of mutations that a woman will have a very, very high risk of breast cancer.